Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen.
Personalised medicine in the broader sense is not a new concept. Patient-specific factors such as weight, age or family history have helpfully informed medical practice for decades, if not centuries. However, revolutions in genetics, molecular biology and computer science are allowing us to place patients into new groups previously invisible to the medical eye and to tailor treatment on an even more individual basis. The consequences for medical decision making, which inevitably balances risk and cost against potential benefit, are monumental. My guest today is Dr. Emma Davenport, who leads a research group at the Wellcome Sanger Institute. Dr. Davenport's research aims to understand heterogeneity in patient responses to inflammatory diseases and their treatment, including sepsis, by bringing together genomics and gene expression data. Dr. Davenport, it's a pleasure to have you with me today.
Thank you very much, it's lovely to be here.
So, what do we mean by personalised medicine today?
So I think today personalised medicine is all about trying to give the right drug to the right person at the right time.
Moving on from that, what sort of applications can we see now?
So I think what's really exciting with the recent developments in technology is that actually we can start integrating in the most personal of all traits, which is someone's genome. So a genome contains someone's complete set of DNA, and it's the instruction manual for all of the cells in the body. And what's exciting is that as we're beginning to generate more and more of this information, we can start integrating it with clinical data to understand why some people are more susceptible to certain diseases and maybe why people respond in different ways to treatments.
So why are these personalised approaches to drug therapy useful then from a patient's perspective?
So at the moment for a number of diseases, particularly common diseases, there can be quite a broad spectrum of symptoms across a patient cohort. So it can be really challenging to identify exactly which drug might work in the right person at the right time. And this is particularly important for the clinical trial space, where there's still a number of problems in trying to get drugs through the various stages of the clinical trial process. And one of the reasons for that is that they often fail quite late in the stages. And so getting a better understanding of which patient population we should be targeting with certain drugs should then help us to sort of really move forward in terms of drug discovery.
We've certainly seen some quite high profile failures in phase 3 trials for Alzheimer's drugs recently, which are quite expensive. But going back to the genome, does our genome tell us everything we need to know about somebody's susceptibility to a disease or to a drug?
No, so the genome is just sort of one piece of the puzzle, and I think the environment plays, a really huge role in all of this as well. So it's also very dependent on people's lifestyle factors. One of the things that we're particularly interested in my group is actually measuring gene expression or activity of the gene. So this is a bit like a dimmer switch of a light, where it can be off or on or anywhere in between. And the advantage of looking at this is that it is affected both by someone's DNA sequence, but also by their environment. So you can start to capture information about both of those aspects that might have an impact on both disease susceptibility and treatment response.
And can we use this information perhaps to predict the people who are going to respond badly to treatments? There's almost a sliding scale from people not responding at all to actually some quite nasty results.
That's definitely an area that we're very interested in pursuing at the moment, and really in disease areas such as autoimmune diseases, it's still very challenging to know exactly which treatment might work on different people. One of the main strategies at the moment is to try a drug for a certain period of time to see whether or not symptoms improve. But that's obviously then not very beneficial to the patient if they have to spend a long time going through a lot of different treatments, and very expensive as well. So if you can try and use genetic information and then potentially gene activity or gene expression readout to sort of try and determine whether or not someone's really having any sort of response, then that could be really valuable for trying to tailor that treatment to that person.
And then looking again to tailor individual treatments to individual patients, can we change the dose at all?
I think that's also another area that's going to be really interesting to look into, because trying to get the balance between a drug that's effective versus then giving so much that it can become toxic is also very difficult. So, Warfarin is a good example with thinning the blood if you've got a blood clot, but obviously warfarin's also used as a rat poison. So you definitely don't want to go too high with that, but you want to get that balance, and each individual may end up having a slightly different threshold for what's going to be the range in which you get the therapeutic efficacy that you're looking for.
It's often said that everything's poisonous, it's just the level that determines that.
Exactly.
So what do you think perhaps the barriers are to embracing this as a healthcare system, embracing this personalised response? Now that we've had the genome revolution, we should be able to sequence more and more.
So certainly there has been disappointment in the genetics field in general with how slow it has been to then be able to really make big leaps in terms of patient care. I think it's a really exciting area though still, and that we are generating huge amounts of data, but now the challenge is really going to be how to integrate that with the clinical data to really maximise the output that we can get from it. I think that one of the major challenges is that for a lot of common diseases, there will be a lot of variants or positions in the genome that vary across people, but they have very, very small effects. So really sort of trying to understand how these work in combination in a particular individual is going to require really large cohorts of individuals, and then integrating that with extensive amounts of the electronic health records as well.
So moving on from that, do you see more collaborations between different groups in different countries, therefore to increase that cohort size?
I think that's definitely going to be very needed, and also the collaboration across the different research expertise, so having people from the different fields working together, because you really need information in the clinical setting as well as information from the statistical genetics perspective to then try and pull those two together sufficiently well to really move forward.
And there's often been perhaps an accusation that the majority of data from clinical trials and the majority of genotype data is predominantly from people of a European heritage, and actually this is leaving quite a few people out of the genomics revolution. What do you think about this as an issue?
I think that's definitely an issue, and the field are very aware of that and looking for ways to try and improve that. And I think one of the things that we really need to focus on as well is public engagement, so really then trying to recruit populations who historically may not have been involved in trials and really try and reach out to ensure that they are included in research going forward. And there's a number of different groups and endeavours to really try and sort of start diversifying the genetics, because it definitely will be really important to have representation from across different populations.
Do you think it's a question of companies and organisations not seeking out these people, or people not being as happy to be involved from particular groups, or maybe a combination?
I think that there is a combination. Obviously, if you are relying on a local population for your recruitment, then that then ends up sort of having a skew based on where researchers are and the resources that they have to be able to generate these large data sets. So it will require good, strong collaborations going forward to try and reach out. But then there's also a need to sort of really engage with people and discuss what these research areas are really focused on to ensure that people are comfortable with sharing their genetic data for this type of research.
And certainly when it comes to sharing genetic data, there's been a lot of talk about people's privacy in this context, and it's said that the more you anonymise data, the less actually useful in a clinical context it becomes. So how do you think we can walk this tightrope between producing useful genotype data with lots of data from people's lifestyles and their family history, but also maintaining people's privacy at the same time?
It's certainly a tricky balance, and I think that is going to be a big focus of this particular area of research going forward, in particular with large research focuses where, for example, like the UK Biobank, where you've got thousands of individuals participating. If we want to be able to create more of these resources, we need to then make sure that the public are comfortable with sharing their data. And so I think, again, the important thing is making sure that we engage with people and really sort of discuss how data are going to be used and try and highlight some of the benefits as well as what some of the risks are, so then people feel well informed to make that decision. And then obviously there's a huge amount of work being done in general of how best we store this type of data, how we can manage access to ensure that the data is being shared across people who are using it for the purpose that it was collected for.
And I think the Royal London Free got taken to court and they lost because they shared, I think, over 1 million files of patient data with Deepmind in their collaboration, which was found to be unlawful. Do you think setbacks like this are perhaps tainting the view of genomics in the public eye?
It could well be, and I think it's going to be just really important then to try and make sure that future studies are set up where people, when they're giving consent for these studies, are aware of exactly the types of work that their data might be used to support, so that then this kind of case doesn't happen again in future research.
And moving on, if this data is the possession of the person who originally signed up for it, then where do they right stand in removing it from data sets? Under the Data Protection Act, that's allowed, but then these data sets obviously could be skewed underneath that. Machine learning software could suddenly lose all of its training data.
That's a good question, and I think it can be quite challenging when data have already been used for certain amounts of analysis, and then trying to go back and remove a particular individual can be difficult from that. However, often I think people can be comfortable with the idea that it can then be removed from anything future beyond that. And I would hope that, again, with enough communication with people of how their data are being used, that then there shouldn't be too many cases where people then become uncomfortable later if they've already agreed at the beginning to share their data in certain ways.
Perhaps a slightly different question, which is that once we have this information on our genetic susceptibility to disease or drugs, for example, why do you think this is not translating? There was a prediction that it would translate into people changing their lifestyles as a result, but this hasn't really been seen to be the case, even if we can predict stroke risk, for example, diabetes risk.
Yeah, it's true. I think there's quite a lot of research now showing that even with this increase in knowledge of your genetic susceptibility, there's still not a big uptake in change in lifestyle factors. I think one of the big challenges here is that, as we discussed before, often for a lot of common diseases, it's many small changes across the genome which have an impact, and so therefore your risk might be only very slightly elevated or reduced. And so I think for an individual to find that they are 1% more likely to be susceptible to a certain trait, that then becomes quite challenging to then make the decision that you are going to exercise every day or eat very healthily and not drink much alcohol. So I think that is going to be a challenge, but I think where this is more useful is thinking about then the resource within the clinic. So this could be a way for doctors to then prioritise who should be given certain screening earlier. So for example, with breast cancer, if there are women who should then be coming in earlier and more regularly for screening. So you could then try and pick things up at an earlier time point, even if you can't persuade someone to necessarily change their lifestyle before they then get that diagnosis.
And certainly cancer therapy is one area in which there's been a lot of interest in personalised medicine. I was wondering if you could tell me a bit more about perhaps some of the advances in that field.
It's definitely sort of the most advanced in terms of actually already making use of people's genetic information. So there are various aspects now. There is increased screening of the BRCA1 and BRCA2 gene for breast cancer, so people can look and see whether or not individuals carry a variant in those or a mutation in those genes, which then increase their risk of breast and ovarian cancer. And there's also some interesting work now where panels are being developed to look at the gene expression or the gene activity, and again that can then start helping understand people's risk of disease.
So moving on slightly towards some of the work that you do at the Sanger Institute, there are obviously some genes that are heavily associated with conditions, but other ones that only have a more minor association, and perhaps large groups of them coming together to produce maybe a substantial risk. How do you go about differentiating genes that are actually involved in disease processes and those which have just been flagged up perhaps incorrectly?
That's a really good question, and it's a big challenge in the field at the moment. So one of the main techniques as geneticists that we use to try and understand genetic susceptibility is to look at DNA sequence in cohorts of patients versus a control group to see whether there are particular variants or regions in the genome which are at elevated frequency in the patient population compared to controls. While this then tells you something about that association, it doesn't then tell you exactly anything about the mechanism. And so a huge amount of work is now being done to try and figure out how a change in the DNA sequence can then lead to the biology that you observe, which is then this increased risk of disease. Some of the things that we're doing is to look at the gene expression of individuals. So many of the variants that have been associated with disease are in non-coding regions of the genome. So it's actually really hard to understand exactly how they might be affecting the proteins, which are the building blocks. So we're really interested in the regulation of gene expression. So as I mentioned, that gene expression is a bit like a dimmer switch. You could imagine that a variant in the promoter or in another non-coding region that has a regulatory function could help change that dimmer switch, so either up or down regulate a gene. So we are generating large amounts of gene expression data for large patient cohorts, integrating that with the genetic information and the clinical data, and really trying to use that to give us a bit more of a step towards how a genetic variant might be impacting expression of particular genes and therefore having an impact on the biology.
And how do you go about measuring differences in gene expression which are not static, unlike the genome, over time?
So that's a really good question, and what we are trying to do is to take blood from patients at different time points to start giving us an idea of how much gene expression changes over time that's just the noise that you pick up from repeat measurements, and how much is the expression changing really due to like a change in the environment, for example, the disease severity state getting worse.
And I know that your lab does a lot of research into sepsis and people's response to it and the heterogeneity in the population. So I was wondering if you could tell me a little bit about sepsis. What is it? Why is it so dangerous?
Sepsis is a life-threatening condition that occurs when the body is trying to fight an infection, but it ends up damaging its own organs and tissues. One of the big challenges is that there's no single biomarker that can be used to diagnose it, and it can initially look like flu or a chest infection, and it can affect anybody, but it particularly affects the young and the old. So one of the big challenges for doctors is really trying to work out who might have sepsis and how to then make sure that they get treatment very quickly.
I think an article that you wrote recently in the BBC quotes the figure that it causes over 50,000 deaths each year, or it's estimated to cause in the UK, which is an astounding number. So what treatments do we currently have for sepsis then?
Yeah, it's a big number of people, and again the sort of real issue being that because anybody can get it, it's really hard to predict who's then going to have a really severe response. So the main treatment at the moment is to try and give antibiotics as quickly as possible, and then try and support the organs that are failing. So you can also have fluids that can be given and steroids if the individual is no longer responding to the antibiotics. But we know that the sepsis is caused by a dysregulation of the immune system, and one of the big challenges at the moment is that we don't have very good drugs that can really target that dysregulation, and so that's a real focus at the moment, is trying to come up with therapies that modulate the immune system that then might be able to help treat individuals who aren't being able to sort of get better with just the antibiotics alone.
So what sort of immunological treatments are we looking at then?
So this could be, for example, a monoclonal antibody that might bind to a cytokine that's released, because we know that cytokines are important in terms of the immune response to sepsis.
And then, I know, moving back to the personalised approaches, why do you think we haven't seen these kind of improved responses to treatments for sepsis?
So one of the big challenges with sepsis is it's a very heterogeneous disease. So you can take a cohort of patients who have all been designated as having sepsis, but their clinical symptoms can vary quite wildly, and the clinical symptoms alone are not good enough predictors to really highlight the individuals who are most at risk of dying. And worldwide, a third of people who develop sepsis will die, so it's a really sort of high number. So you really need to be able to pick who those people are that require the attention and support. We think this has had an impact on the clinical trial space, because obviously you're then getting a very heterogeneous cohort if you're trying to look for certain outcomes to be able to sort of determine efficacy and safety of a drug. Then you're not going to meet those endpoints if you've got such huge variation across individuals. So we're really interested in how we can use gene expression signatures to better understand why there is so much variation in people's immune response to sepsis.
And delving out for 1 second into just the clinical trial sphere, do you think this has been an issue across the board in clinical trials, in the ones that haven't incorporated genomics, that actually some of the trials that have failed, we might have not picked up on certain actually quite significant responses, but just in a minority of people?
I think that's an area that will be really interesting to try and dig into further, and certainly something that we are particularly focused on. And this idea of really trying to stratify patient groups and making use of genetic information, whether that's DNA sequence or gene expression, to try and pinpoint whether there are subgroups of patients that actually are demonstrating a good response to a particular treatment. And therefore, that's a way of ensuring that drugs might be more likely to then pass through the pipeline into the clinical setting.
So on one hand, we've got gene expression, on the other hand, we've got genomic code itself, and then, of course, the epigenome above that. So how do you go about making the links between those areas?
So we are particularly focusing on the DNA sequence and then looking at how that's associated with changes in gene expression. When you can then add in that epigenetic layer, then that's very interesting for looking at the sort of ways in which the genome is open or closed, and that can help you understand whether particular variants are then having a role in gene regulation. It can be quite challenging to try and build up all of these data sets across the same individuals, because that's obviously the most helpful data set to have. And so one thing that we are making use of is then publicly available data where cohorts, particularly healthy donors, have volunteered and given blood to then try and generate reference data sets too. So while we can't always have this information from the person of interest, we can start integrating in information from other people to try and learn patterns across the genome that might help us interpret it.
And certainly sepsis is or can be quite a rapidly progressing condition, and actually part of some of the recommendations, the so-called sepsis 6, recommend 6 different actions, including IV antibiotics and fluid, within an hour. If we're talking about measuring gene expression for an individual patient, how do we balance up this need for speed with actually perhaps a slower, more precise response in predicting their susceptibility?
This is definitely a really challenging area, and there's a couple of areas where we think this could help. So the first of which is that often it's the GPs who might have to make a decision about whether or not to elevate someone's care to the hospital setting. So if there is a way of trying to measure gene expression much more rapidly, and we can come up with a small gene set that could really flag whether people are on a trajectory towards a more severe response, then that could be a good marker for then sending somebody in for that elevated level of care. However, what's more likely is that this won't take away from the sepsis 6, but it will simply then be that next level after that. So once they've had that intravenous fluids and antibiotics within the first hour, it's then what do you do next? And so that gives you a little bit more time to then try and get these tests run, but it's still within that first day of trying to then pick the right next phase of treatment to give to the people that are most at risk of dying at that point.
So we can obviously, or we hope to be able to divide people up into high-risk groups and low-risk groups. But in 2016, you published a study that identified 2 different sepsis response signatures, which obviously was linked to risk. But what differences did we see between these groups?
So in this study, we looked at gene expression in a cohort of patients in the intensive care unit and found that we had these 2 groups that differed quite dramatically in their gene expression response. And the genes that were differentially expressed were really involved in many different immune functions that gave us a sense that one group that had the poorer outcomes had a much more immunosuppressed phenotype and showed evidence of things like T-cell exhaustion, whereas the second group who had the better outcome had a much more immunocompetent phenotype based on the pathway that we saw.
And in this case, how can we tell the direction of causality here? Is it the T-cell exhaustion that's come first and has increased susceptibility, or is this the bacterial infection leading to changes in the immune system that we can target?
It's an area that we are focusing on now, and one of the key questions we have is whether or not people are cycling between these 2 groups. So one hypothesis is that this might be just a mechanism of trying to regain homeostasis in the immune system, and people are swinging from the immunosuppressed state into the immunocompetent and back again in that effort. Our other hypothesis is that actually genetics is playing a role in determining the way that people respond to an infection, and so it might be that some individuals are predestined to go into one response state versus the other. And so now that we've conducted this large-scale analysis on a large cohort of patients, we'll start digging into more of the functional work and sort of picking a smaller group to then look more closely at the cells that might be driving this response and to see whether or not we can understand what comes first. Of course, this work has all been done in very sick patients who are already in the intensive care. So our next project is working with collaborators as part of the bioresource in adult infectious disease cohort. And that's looking at patients when they are first presenting to the emergency room. So the idea with this is that it will give us an indicator of whether or not patients much earlier on in their disease progression are showing evidence of these signatures. And so then that would help us sort of determine what this trajectory is of how people are getting sick and what are the features that are coming early on in that progression.
And you mentioned earlier that there are currently no accurate biomarkers for identifying people with sepsis. Is this something that you think will change in the future?
It's certainly something that we hope will change, and it may not be that you can find necessarily a biomarker for sepsis, but you might be able to find biomarkers that help to stratify patients and really highlight those who are most at risk and need a greater amount of support. Whether or not that's going to be gene expression or maybe proteins that are circulating in the blood, but we're hoping that our work will at least help to try and identify certain pathways or genes that are then the most likely to follow up in the future.
And do you see a future perhaps in which by the time a patient is even presented with sepsis, they already have analysis of their genome on the medical record before we've even started with this?
I think that's certainly looking like it could be a possibility, particularly with things like Genomics England, where there is this big push to sequence particularly rare diseases and cancer, and then individuals have information about their genome which can be attached to their health records. And I think there's increasing numbers of people who are voluntarily sending off their samples to get their own data back, and they then might choose to share that with their clinical practitioners. So it could well be that going forward, more and more people have information about their DNA sequence when they're in a clinical setting. The challenge, of course, is going to be to really interpret how these very small changes in the sequence really then lead to these different responses and these different susceptibilities.
I've got a rather startling fact in front of me relating to speed of response to sepsis, which is that there's an 8% increase in mortality for every hour delay in antibiotic administration, which is enormous.
Yes, it's quite startling with sort of just how much a sort of need for speed is required in this setting. And I think again it's really trying to make sure that people know what the symptoms of sepsis are and how to recognise when someone is very, very unwell and should be then going to hospital to get access to this treatment. But you can of course then appreciate that for a clinician it's very challenging to make that call on who is more sick than someone else if it's deteriorating that rapidly.
Of course. We've seen recently lots of quite high-profile media cases with particularly young people who are previously very healthy, suddenly deteriorating and often dying from sepsis. Do you think in this context we are properly recognising the significance of it as both members of the public and as clinicians?
I think there's still quite a way to go to really raise awareness, I think particularly in the public setting. And while these cases are, heartbreaking to hear about, it has resulted in, I think, a bit of more of an awareness of people recognising that it can happen to anyone. And while it is more likely to happen to those that are very young and those that are very elderly, these cases where it is a generally fit, young, healthy person, it is quite scary that they can then deteriorate so quickly. So yeah, I think there's a lot more to be done though with really trying to remind people that if there is a suspected infection and people are very unwell, then sepsis should be considered.
Of course. And so sepsis is one example of a condition which could perhaps benefit from stratification of patients into different groups for treatment purposes. But are there any other examples that you can think of?
Yes, so another disease area that we're working on is systemic lupus erythematosus, or SLE, and this is an autoimmune disease that is systemic and it can affect virtually any organ in the body. And again, it's a disease that's very heterogeneous, so patients present with a variety of symptoms and you can have different organs involved. So again, it can be very challenging to figure out who requires which treatment when. The other challenge with SLE is that people have a flare and then go into remission. So that makes it challenging as well to know whether or not a treatment is really working or if it's simply the disease progression. So this is another area where we're really interested in looking at the genetics and the gene expression, and again trying to look at that profile in the blood to work out whether or not we can subset patients and highlight those that might be most likely to benefit from different types of therapies.
So what sort of differences are we looking at in this case when we're trying to decide a therapy for a particular patient with lupus?
So there are a number of different monoclonal antibodies that can be used for lupus and other autoimmune diseases, and they tend to target particular pathways. So for example, it might be IL-6, which is one of the cytokines. And so one area that we're quite interested in is trying to understand, could you see whether or not a particular pathway is up or down regulated in a patient, to then figure out which of the sort of spectrum of drugs you have might be most likely to work on them, because their symptoms might be the same as someone else's, but it's a different cytokine that might be elevated in them, and therefore you wouldn't know without looking at that which of the ones is the one to target.
And something perhaps common in research with SLE and sepsis as well is taking samples from the blood. Does this limit in any way the data that you can get from patients?
It definitely is a limitation. One advantage with both sepsis and SLE is that they are systemic diseases that involve systemic inflammation, and so the blood should have some relevance to the disease, and so hopefully we should then be able to see these markers there. But there are other organs involved, particularly in SLE. So, for example, it could be the kidneys. And so it's very likely that there will be gene expression profiles that we can't pick up in the blood that will be then present in other tissues. And I think what will be interesting going forward is then if we're able to collect biopsies from people from other sites. And so sometimes that is a possibility. So, for example, with SLE, with people with kidney involvement, sometimes a kidney biopsy is taken. So if we can then measure gene expression there, we will then get a nice profile of what the immune system looks like in the systemic immune response versus in an organ specific.
And can you see any other parallels with other inflammatory diseases?
Yeah, so we have other groups in our department, for example, working on irritable bowel disease. But again, there is an interest of whether or not the blood might show any features of inflammation. So whether or not you could use the blood to then, for example, find a gene expression or a protein biomarker that might represent what's actually happening at another site. So I think there are multiple instances in which it is going to be useful to try and understand what's happening in the organ versus what's happening in the blood.
And interestingly enough, in 2016, the Society of Critical Care Medicine and the European Society of Intensive Care Medicine actually updated the definition of sepsis, and they moved it away from inflammation as a definition and towards organ dysfunction and organ failure. Has this changed the research environment, or does this reflect a change in the research focus?
I think this reflects a change in the sort of understanding of sepsis and what might be the most important areas to be focusing on. The previous definitions resulted in an even more heterogeneous cohort of patients which focused on inflammation, but you can end up then with patients who have a suspected infection are showing some symptoms but actually don't deteriorate as rapidly as those that have sepsis. So the idea with this is to really try and focus in on the group of patients who have a much higher mortality. So this definition is looking at people who then have a mortality risk of around 10%. So you're really trying to focus in on those that are showing signs of suspected infection, but the organ damage is then really sort of highlighting those most at risk.
Do you think that perhaps these blanket definitions of sepsis or lupus, or even we've seen with asthma as well, which has been divided up into many different patient groups, do you think these blanket definitions are perhaps slightly unhelpful from a perspective because it enforces the idea that these are the same disease?
I think you have to start somewhere, and so it makes sense to start with the clinical symptoms, but it will be really fascinating just now that we have access to the genetic and gene expression data to start working out actually is there more common sort of shared features across individuals. So, for example, in the autoimmune space, particularly in rheumatology, there can be often quite a long period of time before individuals are differentiated into, for example, rheumatoid arthritis or another autoimmune disease. So actually what might be interesting is to then start looking at which gene pathways are being up or down regulated and whether or not those could be used to sort of designate people into these different groups rather than necessarily labelling them as having a particular disease. And that also then might help treatment because you're then targeting particular components of biology rather than necessarily a set of clinical symptoms, which can end up being quite shared across lots of different people.
Gene activity can be looked at from, I guess, a number of different stages, from actual transcription of the DNA in the nucleus to translation into proteins and then activity of those proteins. Which stages in that are you looking at when you look to characterise gene expression?
So we're currently looking at the transcription in the nucleus. So obviously then there's many steps at which modifications can occur. The advantage of looking where we are is that it's very close then to the DNA sequence. So if we're trying to look at a direction of effect between the genetic information having an impact on gene expression, then that's the next sort of closest step that we can look at to try and find those associations. But obviously we don't know that necessarily then translates into a change in the protein level and protein activity. And so what's exciting is that there are many other groups who are now working on different aspects to then try and get that suite of sort of information all the way through from the DNA sequence to the RNA, to the protein, and then finally to the change in the clinical symptoms.
And this looks like quite a lot of data that could be coming through. And I was wondering, there's been a lot of talk about using machine learning and using AI software for data analysis. And I was wondering if that had come to your lab at the Sanger Institute.
We haven't started working on that yet, but I think as the cohort sizes expand, then this is certainly an area that could be of interest, and particularly the possibility of making use of electronic health records. So this is certainly an area where you then have many data points that you can start making use of this type of analysis. The disadvantage there is that the health records can be very sparse, and you have to sort of have a good understanding of potential biases in the data, how data input by the clinicians, and really sort of trying to interpret what it means. So while it is certainly a very exciting area, I think there's going to be a lot of work before we can really see the benefits coming out at the other side.
And even now there's been some exciting work with natural language processing, which can take quite unstructured healthcare data from health records in the form of patient notes and then translate it into something that actually machine learning software can work on. So perhaps that's something for the future. So, well, speaking of the future, fast forward a few years' time, where do you see the field?
I think what we would be really excited to see is being able to measure gene expression in clinical trials so that you can use it as a way of monitoring how people are responding to treatments and trying to then subset individuals based on whether or not they're responding or what their baseline expression is in the first place. And then beyond that, it would be really interesting to then trying to start sort of integrating genetic information with someone's clinical profile to have it as another layer of evidence for understanding what their level of susceptibility to a disease is, and maybe helping to sort of gauge where to start in the panel of treatments that might be possible for them.
Dr. Davenport, thank you so much for joining me today. It's been a fascinating discussion.
Thank you very much. It was a pleasure to talk to you too.
Thank you for listening to the Phager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.